Hi everybody, I hope everyone is well. Welcome to our lesson on Unit 3, in which we are going to be looking at the topic of meiosis. Meiosis is an example of cell division, and we have looked at another type of cell division called mitosis in our previous work. However, in today's lesson, we are going to focus on meiosis. Meiosis, in summary, is a reduction division, and in humans and animals, or some animals, the process of meiosis is responsible for the production of our gametes, so your sperm cells in males and your egg cells in females. And in plants, meiosis takes place to produce a spore in the alternation of generations. So next we will look at the process of meiosis and how it fits into the life cycle of a human being. But before we do that, let us touch on the word haploid and diploid. Diploid means 2 chromosome sets and haploid means 1 chromosome set. And we represent a chromosome set by the letter N. So N by itself simply means we are haploid, and 2N means 2 chromosome sets, so we are diploid. In the case of humans, all human body cells have 46 chromosomes, and all human sex cells, so your egg cell or your sperm cell, will contain 23 chromosomes. If we now take our attention to this image, we can see that the process of meiosis is taking place in your ovary for a female and in your testis for a male. And as a result of meiosis taking place, we can produce gametes, which is essentially your egg cell and your sperm cell. They are haploid, and so meiosis has halved your chromosome number, or in other words, meiosis has ensured that we move from a diploid chromosome state to a haploid chromosome state. And then, through the process of fertilization, the sperm meets the egg and the nuclei of the sperm fuses with the nuclei of the egg. That will result in a zygote. And because we have an N of your sperm cell being added to the N of your egg cell, your new resulting zygote will be 2N. And so the cycle will continue. So we know by looking at the life cycle of a human that every somatic cell in your body will contain 23 chromosome strands from your father and 23 chromosome strands from your mother. And when it comes to the idea of meiosis, we need to talk about the idea of homologous chromosome pairs. So like I mentioned, we have 23 chromosomes from our father in our body cells and 23 chromosomes from our mother in our body cells. But in this image, we are going to talk about just one of each. So we will look at our paternal meaning from our father's side, our paternal chromosome, and maternal from our mother's side chromosome. During the process of meiosis, at the start of meiosis, DNA replication will take place, and each of these chromosomes, the paternal and the maternal, will form a duplicated chromosome, as we can see here and here, which is basically 2 sister chromatids held together at a centromere. So I'll just draw in your centromere. In meiosis, homologous chromosomes need to pair up. But what makes a chromosome pair homologous? Firstly, they have the same size. Secondly, they have the same shape. And thirdly, they have the same location of genes. So for example, if in purple I show that this region is the gene for eye color on your paternal chromosome pair, the gene for eye color on your maternal chromosome pair will also be found in the same location. And these two chromosomes together, your maternal and your paternal, form what is called the homologous pair of chromosomes, or we can simply just say a homologous chromosome pair. So while we are talking about the idea of chromosomes and homologous chromosome pairs, we can bring in the concept or the idea of a karyotype. And a karyotype displays a magnified image of your homologous chromosomes arranged in their pairs from homologous chromosome pair number 1 all the way to homologous chromosome pair number 23. The first 22 pairs of chromosomes code for autosomal characteristics. These are characteristics related to your personality and your behavior and certain other characteristics. The last pair of chromosomes is called your gonosomes, and in everyday language we can refer to them as your sex chromosomes. However, it's just 4% of your X and your Y which determines whether you are a male or a female. The other remaining 96% of genes determine other characteristics. That's just a by the way information. But the most important aspect that we can see when we look at your sex chromosomes or your gonosomes is that they have different shapes. So in a female, we have an XX pair, and in a male, we have an XY pair. And so in females, all of your chromosome pairs are homologous, as we can see in this last pair for a female. Your chromosomes must be homologous, but in a male, they can't be homologous. Your last pair of chromosomes in a male, or chromosome pair number 23, is not homologous because they don't share the same genes and they are a different size. And the idea of a karyotype is also important because we can determine certain chromosomal abnormalities. For example, if there was an extra chromosome at chromosome number 21, we would know that our individual has Down syndrome. So now that we have looked at the idea of chromosomes, which are important to the process of meiosis, we can jump into the process of meiosis itself. However, just like mitosis, before your cell division takes place, interphase must take place. Interphase is a very long phase of your cell cycle, and two significant things happen during interphase. Firstly, we find that your cell builds up a lot of energy. Remember, the process of cell division is an energy demanding process, and energy will be needed. But secondly, what happens is your chromatin network will make a copy of itself. So there are 46 strands, not chromosomes yet, 46 strands of DNA within your cell. And during the process of interphase, each one of those strands makes a copy of itself in the process of DNA replication. We also have the organelles centrioles, which start to play an important role, but we'll talk about centrioles on our next slide. So interphase has taken place, and we can now enter into our process of meiosis. The process of meiosis consists of two stages, meiosis 1 and meiosis 2, and each of these stages is further divided up into 4 stages. which are prophase, metaphase, anaphase, and telophase. However, the numbering becomes very important. The word prophase itself refers to mitosis, the word prophase 1 refers to meiosis I, and the word prophase 2 refers to meiosis II. And that applies to metaphase, anaphase, and telophase. So let's have a look at prophase 1. The first thing we notice is that your nuclear membrane and your nucleolus start to disappear. Fragments of the nuclear envelope are labeled in this diagram, and we can see that they are breaking up, and that's why they are called fragments. Our spindle fiber, as I mentioned on the previous slide, an organelle called a centriole produces spindle fiber, and these are your centrioles. So spindle fiber starts to extend within your cell as your centrioles start to move to opposite poles of your cell. Your chromatin network has also condensed into chromosomes, and those chromosomes have arranged themselves into homologous chromosome pairs. So we can see that we have 6 chromosomes, 1, 2, 3, 4, 5, 6, and they are arranged in homologous chromosome pairs. So there are 6 chromosomes, but there are 3 pairs of homologous chromosome pairs. We find also that at certain regions, your homologous chromosome pairs touch. And that comes to the process of crossing over, which takes place at a chiasma. So the region where they touch is called a chiasma, and the process of crossing over takes place. So we can see that at the chiasma, your 2 homologous chromosomes touch and they can exchange genetic material, and as a result of that, there is genetic variation. That explains why we look similar but not identical to our mom or our dad. So the key important idea on this slide is that your homologous chromosome pairs touch at the chiasma, crossing over takes place, which results in genetic variation, and this takes place during prophase 1. And our next phase is called metaphase 1. And I remember metaphase 1 by looking at the M in metaphase. M stands for middle, and during metaphase 1, your homologous chromosome pairs arrange themselves onto the middle or onto the equator of your cell, which is called the metaphase plate. In addition to that, the spindle fibers from your centrioles attach onto the centromere of each duplicated chromosome within each of your homologous chromosome pairs. So there is an attachment to the centromere on the red duplicated chromosome, and there is also an attachment to the centromere on the blue duplicated chromosome, and together they are a homologous chromosome pair. So next we move into anaphase 1, and during the process of anaphase 1, we find that the spindle fiber contracts. And if we think back to metaphase 1, the spindle fiber was attached to the centromere of each duplicated chromosome of each homologous chromosome pair. So when the spindle fiber contracts, we find that each duplicated chromosome of a homologous chromosome pair moves to the opposite poles of your cell. So in this image, we can see a duplicated chromosome moving to this pole of the cell, and we can see this duplicated chromosome moving to this point or this pole of the cell. And in metaphase 1, both of these duplicated chromosomes were found in the middle region, and together they were homologous chromosomes. But now, in anaphase 1, each of the duplicated chromosomes of the homologous chromosome pair are moving to the opposite poles of your cell. We then move into the next phase, which is called telophase 1. And during the process of telophase, we notice that your nuclear membrane starts to reappear, as we can see by these purple fragments reappearing. And we also notice that there's no longer spindle fiber. So your spindle fiber does not necessarily disappear, but rather it dissolves into the cell. And following the process of telophase is not a phase of meiosis, but rather a cell cycle stage called cytokinesis. Cyto means cytoplasm, kinesis means to split. And so essentially your cytoplasm splits in very loose terms. And as it splits, it invaginates and it moves inwards and inwards to pinch off. And this region that is formed as it invaginates is called a cleavage furrow. And at the end of cytokinesis, we will have two cells formed. Notice that each cell that is formed has 3 chromosomes. 1, 2, 3. And in the top cell, 1, 2, 3. So we have now halved our chromosome number. We have gone from diploid to haploid. Notice as well that in the beginning, we had a complete red and a complete blue chromosome, whereas now there is a red chromosome with a bit of blue, there is a blue chromosome with a bit of red in each of your cells that have been formed. And so they are genetically different to the original parent cell that has been formed. And at this point, if we are talking about animals, we can say that the gametes have been formed. So this can either be taking place in the ovary, if it is a female, or in the testes, if it is a male. And the process of meiosis 1 has now ended. However, the process of meiosis itself is not yet done. Remember, I said that meiosis has 2 phases, and we now move into our second phase of meiosis, called Meiosis II. Meiosis II is essentially mitosis, so I won't spend too much of time on the process of Meiosis II. I would just emphasize 2 things. Firstly, don't forget to add in the letter 2 or Roman numerals 2 when you are dealing with the process of Meiosis II. And also, I would like to touch on anaphase 2. So remember, during anaphase 1, we had a homologous chromosome pair split up into duplicated chromosomes when the spindle fiber contracted. During anaphase 2, a duplicated chromosome splits up into a daughter chromosome. So if we have a look at the diagram on anaphase 2, that is a daughter chromosome. That is a daughter chromosome. And at the end of meiosis II, in other words, after telophase 2 and cytokinesis has taken place, we have produced 4 genetically different cells. And in the case of humans, we can say that we have produced 4 genetically different gametes. They are genetically different because of the process of prophase, when crossing over took place, and they are also genetically different as a result of metaphase 1, because when they lined up, and by they I mean the homologous chromosome pairs lined up on the equator of the cell, they lined up independently. So that also contributed to genetic variation. And that brings the process of meiosis to an end. Lastly, let us touch on the concept of chromosomal abnormalities. So earlier on in this video, I mentioned that we can have Down syndrome occurring in an individual, and that's a chromosomal abnormality caused when we have an extra pair of chromosome number 21. But how does that actually take place? It takes place in a process called non-disjunction. And non-disjunction basically means that your homologous chromosome pair fails to split up into its duplicated chromosomes during meiosis I, or during meiosis II, your duplicated chromosome fails to split up into its daughter chromosomes. And as a result of non-disjunction, we can have many chromosomal abnormalities taking place. For example, Down syndrome. But we can also have other disorders. For example, Klinefelter syndrome can also take place. So that brings our unit of meiosis to an end. I hope you enjoyed this video, and thank you so much for watching. I will see you soon.